Hello, students. So today we are going to start the topical past papers of chromatography A2. So let's start from the chapter from the question number one. So that is use the data booklet to draw the structure of tripeptide. So tripeptide means that there will be three amino acids, and obviously you can see it like alanine, serine, and glycine showing its peptide bonds in full. So obviously you have to show the peptide bond. First of all, you need to know that what is peptide bond actually. A peptide bond is like C double bond O, N, and H. Okay, and it is formed by the reaction of the carboxylic acid with that of the amino group. And you know, our amino acids contain both of these groups, carboxylic acid and amino acid, so they can form peptide bonds and they can form polypeptides, obviously, you know. So, for this purpose, we need a data booklet to draw the structure, obviously. So, alanine, like there are some, you can see alanine is containing the R group is replaced by CH3. In glycine, it is replaced by hydrogen, and in serine, it is replaced by HOCH2. So, obviously, we have to draw accordingly. Okay, now if I just draw, you can say this is a carbon, and you know there are three bonds are like dissimilar because alpha carbon is just attached to one hydrogen, one NH2, one CH. Only the difference between the different amino acid is the R group. Okay, so we can draw it like CH, and obviously I have to draw a peptide bond. So this will be for example, this is for alanine, and you must I need to know that your sequence should be accordingly. Like first of all, you have to draw alanine, then serine, and then glycine. If you change the sequence, then the question will be wrong, obviously. So that's why, for example, this is like this, and in alanine, the R group is CH3, obviously, and let's say this is NH2. Okay, now if I just change the color for serine, so it will be like this is the NH of serine, and serine, as I told you, that it's like R group is replaced by CH2 OH, and then you can say here is a carboxylic group, and it is bonded with another I can change the color for the glycine. So glycine is obviously this is amino group of the glycine, and this is CH. And just to show you the different R groups, I am writing hydrogen over here, and then you can say it is COOH. So this is our tripeptide, and you can clearly see the peptide bond in it. Okay, so in the three amino acids there will be two peptide bonds, obviously. So these are, you know, this is one peptide bond over here, and this is another peptide bond over here. Okay, and there are different R groups, and the sequence should be same, obviously. Now, the second thing is that you have to calculate the relative molecular mass of alanine, serine, and glycine. So, for that reason, you can just count the number of carbon, number of hydrogen atom, and all that. You know how to calculate the molecular mass. It's so simple. So, there are how many carbons in it actually? It contains, we can count them like 1, 2, 3, 4, 5, 6, 7, 8. Okay, there are eight carbons in this structure, and then hydrogens are actually, I have counted already, so it's like H15, and then nitrogen is like one, two, and three nitrogens. So nitrogen are three, and how many oxygens are there? One, two, okay, three and four, and then five. So oxygens are five in this structure. So now you, if you just calculate it, so its MR is actually 233. Okay, so this is, you can calculate it. Now, the next thing is electrophoresis can be used to separate mixtures of amino acids and peptides. A mixture of the tripeptide, alanine, serine, and glycine, and its three constituent amino acids, was subjected to electrophoresis in a buffer at pH 11. Okay, so you have to draw the structure of serine at pH 11. Okay, so how can we obviously I will write it. Basically, if you just look at the uh, Okay, so the next one is electrophoresis can be used to separate mixtures of amino acids and peptides. So a mixture of tripeptide, alanine, serine, glycine, and its three constituent amino acids was subjected to electrophoresis in a buffer at pH 11. So you have to draw the structure of serine at pH 11, obviously. So it's a basic pH, you know. So if I just draw a zwitterine, so you will see that like this is CH, okay, and this is containing CH2OH. Let's say this is COOH, or obviously I'm writing the zwitterine, so it will be COO negative, and it will be NH3 positive, okay, NH3 positive. So if I will add some basic, I will add it in some basic medium, obviously. So its pH is 11. Obviously, it is basic pH. So it means that there will be a more concentration of OH negative over here. And what's the function of base? It abstracts the proton, obviously. So from where it can take the proton? Obviously, from NH3 positive, it can take the proton. Okay, so it will take the proton from NH3 positive and it will form NH2 CH CH2OH, and this is COO negative. So at pH 11, this will exist in this form. Okay, so this is at the pH 11. This will be its structure. Okay, so now the next question. Uh, at an at the end of the experiment of the following results were seen actually. So first of all, you have to identify the components that are labeled F2I in the above diagram. So this is the electrophoresis, instrumentation. So F is obviously it's a DC power supply. So it's a DC power supply. Then if I talk about the G, obviously, so it's anode, or you can also write as positive electrode. And if we talk about H, obviously it's a negative electrode or it's a cathode. Then I is actually, uh, you can say it's a filter paper that is soaked in a buffer solution. A filter paper soaked in buffer solution. Now, uh, suggest the identities of the species responsible for spot P and for spot S. Okay. So, as we have already seen, different type of like glycine, alanine, serine, glycine, and obviously tripeptide of those. So, a spot P is actually glycine. And why it's a glycine? Because if you can see in all of the three structures like serine, glycine, alanine, glycine has the least molecular mass, obviously. So that's the reason it's like obviously um, here will be glycine because their rate of movement, it depends upon their about their charge and their molecular mass, obviously. And if we talk about the spot S, as you can see, spot S is like it has traveled least, obviously. It means that it will have the highest molecular mass, obviously. So it will be a tripeptide, no? That's why a tripeptide will travel least. Okay, so that's why it is our uh, alanine, serine, and you can say glycine. Okay, so I have to write the explanation also. So because glycine is smallest, glycine is smallest, so travels fast. Tripeptide is largest, so travels least. Is the largest, so travels least. Okay, next. State the reagents and conditions that are needed for converting the tripeptide into three constituent amino acids. Obviously, this is like we will hydrolyze them. Okay, so through hydrolysis, in the acidic or basic medium, we can we can break the peptide bond and we can convert into the constituent amino acids. Obviously, so you can either heat with the H3O positive, or in another way you can write as heat with OH negative but aqueous, obviously, because you know there will be hydrolysis, so aqueous conditions will be there, and the name, the type of reaction, obviously it's hydrolysis. Okay.